So with this, I'm happy to introduce our next speaker, Vladimir Kata from University of Geneva. Vladimir's lab's been doing some fantastically precise cell signaling studies across different model organisms, and today he's going to talk about mutational landscape and GNAO1 associated disorders.
Thank you very much for the introduction. Thanks a lot to the Bow Foundation for organizing this event. I'm very happy to be part of it. I'm even happier now because my luggage is finally delivered. So you see this scheme shows like a workflow of experiments in my lab dedicated to GNAO1. So we start with patients with a specific mutation. We try to decipher at the molecular and cellular level how these mutations lead to aberrant signaling. By doing so, we also establish the assays which can be used for the high throughput screening. Whatever hits we identify, we validate them in the animal models which we create in parallel, and finally we go back to the patient. But actually, what I want to dedicate most of my attention today is the genetic nature of the mutations which affect the patients. So the first paper was published in 2013, and since then there are many more papers published. And basically what we know is that we are dealing with the de novo, in most cases, heterozygous dominant mutations. Well, actually, there are different types of heterozygous dominant mutations or mutations in general. And this was introduced by the guy called Herman Miller, who got the Nobel Prize for his pioneering work on genetics. And the mutations he identified are called morphs. So there are 5 classes of mutations or morphs. The first one is what we call a null or loss of function. Miller called it amorph. Then there are hypermorphs, partial loss of function. Then there is antimorph, which is the dominant negative. Then there is a hypermorph, which we call a gain of function. And finally, what is often ignored in medicinal genetics is the 5th category, which is called a neomorph. A neomorph is a mutation which leads to a new function which did not exist before. And the analysis of the GNAO1 patients in this disease spectrum leads us to conclude that basically you find the intermingling overlap of the amorph, hypomorph, and antimorph alleles on one hand, and they lead to the mild spectrum of the disease, typically having dystonia, but also you could have dysarthria, Parkinsonism, intellectual delay, and so on. And then there are stronger mutations, which, as I will show you, are neomorphs of 2 different types. There is a neomorph A leading to the NEDIM, which is the movement disorder, severe one without epilepsy, and neomorph B, which is DEE17, so developmental and epileptic encephalopathy. So I want to share with you our work of the recent years on how we decipher these mutations and identify them. So on this slide, I show you the structure or the scheme of G-alpha-O in the complex of the G-beta gamma, and I map here the mutations which belong to this first category, first layer. So we don't know actually whether they're really loss of function or partial loss of function or dominant negative. I think there are all of the 3 at the same time. To give you some examples of these mutations which lead to the milder spectrum, so there is this famous intronic mutation which leads to the appearance of a alternative acceptor site for the splicing, which leads to the isoform which has 2 amino acids which are inserted in frame, more or less in the center of the protein. It's located here. And this mutation is seen quite often in the GNAO1 population, and in the patient we studied, it's actually the only Swiss patient identified so far. So he is currently 21 years old. He is doing largely fine, except for he has a slight dystonia and a slight delay, but really slight. At the molecular level, we have characterized this mutant. It has a bit increased GTP uptake, a bit reduced GTP hydrolysis rate. It loses the interaction with RGS proteins, as GNAO1 mutations typically do, as you will see, and it has normal localization, normal interactions with G-beta gamma and GPCR coupling. So we conclude that this mutant is either hypomorph or maybe at the same time a dominant negative. Then there is another mutant, actually 2 mutants, which we characterized recently with Moritz Thiel. the mutations are located in the alpha, in the N-terminal alpha helix of G-alpha O. And what we showed is that these mutations lead to proteins which are fine in all possible measurable aspects, except for they lose the ability to interact with the G beta gamma subunits. So in this regard, they're also partial loss of function and possibly dominant negative, but in a different manner than the mutation I introduced before. And this leads to a different phenotype. Then there is a mutation in the girl present here, sitting in the audience. It's a mutation found in the Spanish cohort, which is a deletion of one particular amino acid, isoleucine 344. And you can see that the manifestations are mild again. The disease is late onset. And what happens actually with this mutation is that the variants are fine in all measurable aspects, except for they lose the capacity to couple to GPCRs. And this is because the C-terminus of G-alpha O is actually important for the interaction with the GPCRs. And when this deletion occurs, there is no coupling anymore. And in this regard, this is again a dominant negative mutation, but in its own manner. So these mutations, and there are many more, of course, described by other clinicians and researchers in the field, they fall into this first layer of the clinical severity of the disease. And this would also include the deletions or truncations or early stop codons. But then, what about these neomorphs? So the neomorph A or Nedim variants of those which we studied in my lab, they are plotted here. And the neomorph B or the 17 mutations which we studied in the lab are plotted right to the right. You can see that they are quite intermingled, but so what is special about them? So we have done a lot of layers of analysis at the molecular and cellular level. So one is the analysis of how these variants are capable to deal with GTP, because you know we are dealing with the G protein, which is typically inactive when it is bound to GDP, which is activated when the GDP exchanges for GTP, and then it is deactivated when the GTP is hydrolyzed back to GDP, and this reaction can be catalyzed by RGS proteins. And we wanted to know what are these rates of GTP uptake and hydrolysis. For this, we used purified proteins and fluorescent guanine nucleotide analogs. And when we look at several mutants, you can see that the rate of the GTP uptake is dramatically faster for the mutants than for the wild type, by about 2 orders of magnitude. At the same time, many mutants, including these 3, which are among the 3 most frequent ones, they lose the ability to hydrolyze GTP. So the wild type curve would look like that, so there is an increase when the GTP is bound, but then the fluorescent drops. This is when GTP is hydrolyzed. But the mutants, they take GTP up and essentially don't hydrolyze it. And this is the K-hydro reaction rate, which is decreased by about 2 orders of magnitude. And actually, we studied a whole bunch of variants, and this is a more or less uniform representation for the severe mutants. They biochemically behave as if they are constitutively GTP bound, because they take GTP up much faster and they fail to hydrolyze it. The next thing we wanted to see is, again systematically, is how the mutants localize in the cells. So G-alpha localizes to 2 spots in different cell types, including neurons, the plasma membrane and the Golgi. And what we find is that several mutants lose the plasma membrane localization or decrease it to a certain strong level, but many others retain both plasma membrane and Golgi localization. And what appears is that the mutations which lead to epilepsy manifestations, they with just one exception we have found so far, they molecularly lose the plasma membrane localization, while those which lead to the Nedim phenotype, they retain the plasma membrane localization, and both retain the Golgi localization. We also wanted to study how the mutants interact with different partners in the cell, such as G beta gamma. And here we see that by the pull downs, but also by the Brett assay, we see the general feature. The D17 mutants often have reduced G beta gamma binding, which correlates with their reduction of localization in the plasma membrane. We have also studied how these mutants coupled to GPCRs using actually 2 independent assays. And these studies show that different mutants coupled to the neuronal GPCRs to the extent which is reduced in a different manner depending on which GPCR you study and which mutant you look at. So some interactions, couplings can be very much defective, others are more or less at the wild type levels. Another cellular partner we want to look at is RGS-19, or we also studied other RGS proteins. And here came the surprise, which brings me back to the biochemical aspect of the work. So in the pull-down assays, the wild type G-alpha-O interacts with RGS-19 to a measurable level. The classical non-pathogenic constitutively activated mutant, which is always in the GTP state, interacts with RGS-19 like crazy. But the mutants actually lose to a strong extent the ability to interact with RGS-19. And this feature is true for the whole panel with some few exceptions. So this is a very general feature. So here comes a certain contradiction. Biochemically, these mutants are constitutively GTP bound because they bind GTP much faster and they cannot hydrolyze it. Yet they do not interact with the partner, which should interact with the GTP loaded form. So how does it work? The explanation to this paradox, we believe, lies in the fact that the mutants actually fail to adopt the activated conformation after interaction with the GTP. So they take GTP up, but normally this should have triggered a structural rearrangement which makes the protein activated. But this is not happening in the mutants. So these are molecular dynamic simulations. And actually what I want to show you here is this particular catalytic loop where the glutamine 205 is located. And this loop is actually displaced in this mutant. But what is cool is that this displacement is confirmed by the cryo-EM data, which we just start to obtain for the same mutant, E246K. So basically what I want to show you here is that this loop and the catalytic loop and the alpha helix which follows, the model shows the structural crystallographic data, but actually this loop is not resolved in the cryo-EM. Everything is resolved, but this region is not. So actually this suggests now that not just by molecular dynamic simulations, that in the mutants this region is not folded or is very flexible. And this brings us to identify the partners which are, in my view, the most exciting, at least of those which we studied, which are RIC8 proteins. So RIC8 proteins, they are chaperones for G-alpha subunits. There are two of them, RIC8A and RIC8B. RIC8A is a chaperone for G-alpha IO subtype, but also G-alpha Q subtype of G-alpha subunits and G-alpha 12-13 subtype, while RIC8B is exclusively responsible for the chaperoning of G-alpha S-OLF subtype. And since we found the data which suggested that the proper structure of G-alpha-O is not adopted in the mutant proteins, we suspected that maybe this should be reflected in the interactions with RIC8s, which normally for the G-alpha-O wild type are there for the transient interaction. So as soon as the G-alpha-O is synthesized and is still empty of the nucleotide, RIC8 comes in, helps it fold, help it bind its first guanine nucleotide, and then goes away. And actually what we find is that the wild type G-alpha-O indeed interacts very poorly with RIC8A, indeed because the interaction is supposed to be very transient. But the strong mutants which we have studied, all of them, except for C215Y in this panel, they gain the massive, dramatic interaction with the RIC8A. And what is more, they actually relocalize RIC8A. So normally RIC8A is a cytoplasmic protein, and this doesn't change when you overexpress G-alpha or wild type. But if you co-express the mutants, you can see that they draw RIC8A from the cytoplasm to the Golgi. What is even more surprising is that the pathogenic mutants of G-alpha-O, they gain the interaction with RIC8B, which, as I told you, is a totally foreign chaperone for G-alpha-O. But here the situation becomes more interesting. So wild type does not interact with RIC8B at all, and among the mutants, some interact very strongly, others just a little bit or not to significant levels. And actually, you can see the very clear grouping. So the epileptic mutants, they all gain the interaction with the RICADE-B, while the NIB mutants either do it to a low extent or not at all. And actually, we can find a perfect correlation between the disease severity, which we measure as the disease onset. We're not clinicians, we have to choose an easily accessible measure from the literature, which is for us the disease onset. And we can see that among 15 or 16 mutants we studied, there is a very significant correlation between the disease onset and the RICADE-B interaction strength. And the same is true for the Golgi relocalization. So the mutants which interact with RICADE-B, they also relocalizes to Golgi. And this correlates with the disease onset. So basically, my message is that the message which is painted with a broad brush, because there are many subtleties which complicate the picture, but when painted with the broad brush, I want to conclude that there are 3 layers of mutations in GNAO1 encephalopathy. The first layer, which leads to the least severe manifestations, are these loss of function, partial loss of function / dominant negative mutants. The next layer is the neomorph A, which leads to much more severe early onset movement disorder dominated clinical manifestations. And the third layer, the most significant severe one, is the developmental and epileptic encephalopathy with the early onset of movement disorder and epilepsy. Now, I just show this schematic slide out of this work of ours. For the sake of time, I don't show you the experimental data, but it's all published. Basically, we believe that these neomorphic interactions with RICADE-A and B proteins impact the whole network of GPCR and G-alpha signaling within the cell. And we have data which support that. So imagine in a cell, you would have multiple G-alpha subunits which are produced. And each of these G-alpha subunits needs RICADE proteins A or B for the proper folding and ultimately for the proper functioning. But when you have the mutants, the patient cells where this neomorphic mutant is expressed, this mutant basically outcompetes for the RICADE-A and RICADE-B interaction with the wild type, not just G-alpha O wild type, but wild type G-alpha I, G-alpha Q, G-alpha OLF, G-alpha S, and so on, depending on the neomorph A or neomorph B. And this leads to decreased levels of these proteins and decreased signaling by the respective GPCR pathways and complicated phenotypes. So there are 16 G-alpha subunits encoded by our genome, which are grouped into 4 subclasses. So 3 of them, as I told you, are RICADE-A dependent, and 1 represented by 2 G-alphas, G-alpha S and G-alpha OLF, are RICADE-B dependent. So basically, in this neomorph A group of mutations, the RICADE-A dependent G-alpha subunits suffer. I put a cross here. This is an exaggeration. Obviously, they are not gone. They are decreased to different extents, and their signaling is affected. While the even more severe group, RICADE-B, which involves neomorphic interactions with both RICADE-A and RICADE-B, it affects the whole family of G-alpha subunits, including those which are RICADE-B dependent and those which are RICADE-A dependent. So, does it have implications for how we can or should treat the disease? Potentially, yes. So we have performed a high-throughput screening in order to identify small molecules out of a library of approved drugs which could restore the aberrant GTPase activity of one of the mutants, E246K. And this led to identification of zinc in the form of zinc salts or zinc chelators, which actually recovers this defective GTPase activity, which works for several G-alpha subunits. So there is a concentration-dependent recovery in this K hydrolysis, bringing it towards the wild-type levels, although not quite reaching it. At the same time, the wild-type G-alpha O is not at all affected by zinc. So the atomistic mechanism how zinc works at least based on our molecular dynamic simulations, is that this catalytic loop, which I mentioned earlier, which is displaced and highly flexible in the mutants, is brought actually back. This is the distance from the gamma phosphate of GTP and this glutamine 205. And you can see that it is quite distant in the mutants, but upon substitution of magnesium for zinc, this distance goes back to the wild type levels. And we believe this is how the restoration occurs. The cell interactions are restored by the zinc. The implications for the disease treatment is that in the very recent paper, which is now in press, together with Moritz, he will talk more about it because this is the first in-human zinc trial. We have studied the panel of GLFO mutants, and we show that they actually fall into at least 3 different classes in terms of their biochemical responsiveness to zinc. There is a class which is not sensitive to zinc at all. Like wild type, there are variants which do not respond to zinc. And these are the wild type, these are the variants which actually belong to this first layer of the clinical severity. These are the non-neomorphic mutants. These are the mutants which are either loss of function or dominant negative or both. But then the neomorphic mutants respond to zinc. And we have shown in the Drosophila model of the disease where the motor activity of the flies having the pathogenic mutation is defective and the lifespan is decreased in these flies. We can show that zinc treatment of these flies recovers the motor capacity of the flies to the wild type levels, at least in some settings, and it significantly recovers the lifespan, although not to the wild type levels. We have done the safety study of the zinc supplementation in mice. It was important to test it in the mouse PAPs because it was studied in adult mice but not in the mouse PAPs, and there is very good safety. And then we studied zinc treatment in the C215Y mouse line which we have. I remind you, this mutation by itself does not respond to zinc, but we thought still this probably is a good model to study the safety of zinc in this animal model of the disease. And it is fully safe. Actually, in some assays, zinc treatment led to the improvement in the behavioral assays, but this was genotype independent. So zinc is safe in the mice. And this led to the first in-human trial where very significant improvements, although not curing of the patient with the G203R mutation, have been observed. I skip this because I know Moritz will be talking more about it. But these findings now lead to the clinical trial, which we will start in Germany in July. And actually, there is a clinical trial which I contributed to in terms of convincing the clinicians in China to start it with interesting data. Safety is good. This is the most important thing. And some patients report improvements, but this we need to wait for the completion of the study. And with this, I terminate my presentation. So these are people in my lab who were involved in this work, and I'm thankful to multiple collaborators across the globe, mainly clinicians, but not exclusively, with whom we study individual patient mutations. And I thank the funding bodies. Actually, I realized that now I should also add GNAO Familia because we just got a grant from them for one of the research projects. So thanks for your attention.
Thank you, Vladimir, for a very exciting talk. We've got time maybe for one question.
Thank you, Vlad. Nice presentation. I was just wondering, because we see differences when you overexpress GLFO and possibly also RIG8. So I was just wondering, does this also reflect the endogenous situation of a patient?
You're asking
whether the neomorph function you see, whether that's also there in the patient, because this is all overexpression based systems. So,
well, we have introduced the several mutations in the through the knock-in in a cell line, neuroblastoma. Neuroblastoma cell line, and we see the same neomorphic interactions. But this is still, of course, only an approximation to the situation in patients. But my answer is yes. The data which we have do indicate that the phenotype, the mechanisms which we discovered, they are also happening in the patients. Whether these are the only things which lead to the pathogenesis, I don't know. I would say these are the major things, but maybe not exclusive.
Okay, thanks.
All right, thank you very much, Vladimir.